Welcome to Unit 3. Here we will look at DNA replication. The ability of a cell to replicate and produce daughter cells is a fundamental requirement for homeostasis. This is defined as the maintenance of a stable and balanced state. Cell division is required for development of an organism, that is, for the renewal of stem cells and the differentiation into cells with specialized functions. As well as that, there's a need for cell division to replace dead or worn out cells. Cell division must ensure that the genetic material, that is DNA, is replicated correctly and transferred faithfully to daughter cells so that they can carry out their specialized functions via gene expression. Cell division is usually triggered in response to external growth factors that signal through their cognate receptors that are embedded in the plasma membrane of the cell. Here the ligand-occupied receptor triggers complex signal transduction cascades that leads to cell proliferation and division. For a cell to divide, it must go through a number of distinct phases known as the cell cycle. In the G1 phase, a checkpoint occurs to ensure that the cell is of the correct size. In the S phase, the DNA is replicated, and this is the subject of this part of the lecture where we look at DNA replication in more detail. The third phase is the G2 phase, and again this is another checkpoint to ensure that all the cellular components are in place so that cell division can occur. The last phase is known as M phase or mitotic phase, and this is whereby the cell divides in two and the DNA is separated equally into the two daughter cells. A major advance in DNA replication occurred in 1953 when James Watson and Francis Crick published their work on the structure of DNA in the journal Nature. Here Watson and Crick built a model which they proposed corresponded to the structure of DNA. They proposed that DNA was a double-stranded molecule that adopted a helical structure, where each of the DNA strands ran in opposite directions. And at the end of their short paper in 1953 in Nature, they said, "It has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material. Hence it laid the foundations for understanding DNA replication and cell division." In a subsequent publication that year, Watson and Crick outlined their hypothesis of how DNA replication occurs. Here they stated, "Now, our model for deoxyribonucleic acid is, in effect, a pair of templates, each of which is complementary to the other. We imagine that prior to duplication, the hydrogen bonds are broken, and the two chains unwind and separate. Each chain then acts as a template for the formation onto itself of a new companion chain, so that eventually we shall have two pairs of chains, whereas we only had one before. Moreover, the sequence of the pairs of bases will have been duplicated exactly. And indeed, this hypothesis they outlined was proven to be correct." What was not clear at that time, however, was how the process of DNA replication took place. Three models of DNA replication were suggested at that time. The first was known as conservative replication. Here, the parental strands of DNA separate and newly formed daughter strands are copied. The parental strand then reforms, shown here. The daughter strands are shown here in light blue. The second model of DNA replication was the semi-conservative replication model. And in this model it was proposed that each of the two parental DNA strands separate, shown here, and act as templates for the daughter strands to be synthesized. The third model was known as dispersive replication, shown here, and in this model it was hypothesized that both sets of DNA have double-stranded segments of parental DNA and newly synthesized DNA which is interspersed with the parental DNA. The mechanism of how DNA replicates was finally solved in 1958 when Matthew Michelson and Franklin Stahl grew E. coli bacteria in heavy nitrogen. They subsequently switched the cells to a lighter nitrogen after one round of cell division and followed the cells. They conducted experiments to conclusively demonstrate that DNA was replicated via the semi-conservative mechanism. That was the second model we spoke about on the previous slide. And here I will direct you to a YouTube video which outlines the process of how they conducted their experiment and how they interpreted it. So you're directed to watch this. We now know that DNA replication occurs at origins of replication in both eukaryotes and prokaryotes. These are located at various points along the DNA molecule, shown here and here and here. There's 3 points in this diagram which are origins of replication on the eukaryotic chromosome. Here the DNA strands separate and enable the replication machinery to produce a copy of each parental strand, shown here. Replication can proceed in both directions, so to the left and to the right. As the new daughter strands are formed, shown here in light blue, replication bubbles form, and you can see these bubbles forming here. And these can be visualized by electron microscopy. You can see these bubbles forming here along the DNA. The replication bubbles eventually fuse and the synthesis of the daughter strands is now complete. Here you can see in dark blue the parental DNA strand and in light blue the daughter strands. A similar mechanism also occurs in prokaryotic cells as shown here in this diagram. And again, these replication bubbles can be seen by electron microscopy. There are several enzymes and proteins that are involved in DNA replication. This includes helicase enzymes, which are able to untwist and unwind the double helix prior to replication. Once the DNA unwinds, another family of proteins known as single-strand binding proteins bind to the DNA and prevent the single-stranded DNA from reattaching. Thirdly, there's a family of enzymes known as topoisomerases, and these are usually found up ahead on the double-stranded DNA, and what they do is that they relieve tension in the DNA by breaking, swiveling and rejoining the DNA. The replication of DNA requires 5 to 10 RNA nucleotides, which are also known as RNA primers, to be deposited at the origins of replication. This step is catalyzed by an enzyme known as primase. There is a significant difference between transcription and DNA replication in that the transcription of DNA does not need RNA primers, whereas DNA replication does require an RNA primer. Enzymes called DNA polymerases catalyze the incorporation of DNA nucleotides onto the RNA primer. DNA synthesis proceeds in a 5' to 3' direction. Now there are differences between eukaryotes and prokaryotes in the types of DNA polymerases that catalyze replication, but the mechanism is similar. For example, in E. coli, which is a prokaryote, there are several different DNA polymerases, but only 2 appear to play major roles in replication. That is DNA polymerase I and DNA polymerase III. The situation in eukaryotes is more complicated, with at least 11 different DNA polymerases discovered so far. However, just for prokaryotes, the general principles are the same. The rate of elongation is about 500 nucleotides per second in bacteria, and it is about 50 nucleotides per second in human cells. Therefore, bacteria can synthesize and replicate DNA about 10 times quicker than what can occur in human cells. Please look at these links for more information relating to DNA replication. So DNA replication is a highly accurate process, but mistakes can occasionally occur, such as a DNA polymerase inserting a wrong nucleotide. About 1 in every 10 to the 5 nucleotides incorporated into the newly synthesized DNA strand will contain an incorrect nucleotide. However, proofreading occurs where the new nucleotides are checked against the template DNA strand. Most of the mistakes made during DNA replication are promptly corrected by DNA polymerase itself, as it can proofread the base that has just been added. Some errors, however, are not corrected during replication, but are instead corrected after replication has been completed. This type of repair is known as mismatch repair. In mismatch repair, the incorrectly added base is detected after replication. Mismatch repair proteins detect this base and remove it from the newly synthesized strand by nuclease action. The gap is now filled with the correctly paired base. Some environmental agents such as UV radiation can catalyze pyrimidine dimers. This can happen where the UV radiation causes two thymines which are on the same DNA strand to form a bond. This can interfere with DNA replication and gene expression. In most cases, these pyrimidine dimers are repaired by nucleotide excision repair, and below here you can see that the bond has been removed and the error has been corrected.